Thank you, everybody, for joining us for another IMAG MSM working group on multiscale modeling and viral pandemics miniseries. This working group is co-organized by James and I, and also, of course, it wouldn't be possible without technical and organizational support by Jim, Sluka, Lorenzo, and Joel. As always, we are visible, trying to be visible on multiple media, including our Slack channel, Twitter, wiki page. There will be also a new website coming up soon. Also, we're on LinkedIn and YouTube, where you can see all of these presentations either live or as well as in a sort of archived fashion, so you can go back and view any of these presentations from the past. Please follow us on Twitter and also help us share any announcements that we make there. Are there any announcements from the larger group? No, okay, thank you. We have several publications that have been added by members of our working group. You can see them here. And if you'd like to check them out in more detail, they should be on our wiki page and the website. So we're now at 30 publications that have resulted from the collaborations and work and discussion in our subgroups and the main working group. So this is amazing to see the productivity growing out of this working group. We will have a presentation on October 20th that will celebrate our 2-year anniversary of this working group. And so if you would like to contribute to the content of the presentation, please email Jim, and you can also, we can send you a link, this link that's available here on the slide here. We can send you a link to the presentation to see what we have at this moment. So here we have a schedule for the upcoming mini seminars. So we are pretty booked and excited to have all these speakers. And we are looking to fill the schedule for 2023. So if anybody has any suggestions for speakers, please send them our way. So it's my pleasure to introduce today's speaker, Liane dos Santos Canas, who will talk to us about her research from early detection to disease profiling. How can machine learning models detect early signs of COVID-19 and can be used to profile post-COVID syndrome? So thank you so much for joining us, Liane, and we look forward to your presentation.
Thanks for the opportunity of presenting today. And the work that I will be presenting is actually a collaboration between King's College London and Zoe Health. And yeah, as introduced, I will speak about how can we actually try to detect COVID-19 and also try to profile the disease when it occurs on its long form. As most of us know, COVID-19 is a respiratory illness, as been affecting lots of people worldwide, causing not only hospitalization, but actually death as well, and long-term disease. And what we try to do is trying to build a population-based study that would allow us to acquire data to not only diagnose, but actually to keep monitoring the patients and trying to understand what is going on with them and with their disease and with their symptoms. To do it so, we made use of an app where the patients could register, patients or other individuals that were willing to participate, and they would report any daily symptoms. So with this app, which includes not only UK, but also US and Sweden, we were able to reach more than 4.7 million contributors, which contributed daily with any symptoms that they could feel, and also they reported any tests, both negative and positive, and they provided also information about the vaccination. As you can guess, this is a huge amount of data, which brought us some technical challenges. The majority of them were related to the need to do some data curation that would be robust and reproducible, not only across our studies, but also for someone that could assess the data. On top of that, since we were using such amount of data, that also required huge amounts of memory to process this. So in order to create these pipelines for data curation that were robust across our studies, we, or part of our team, developed an open source software. You have the link here in GitHub called etc. That it works as a pandas-like and would provide the development of these pipelines. So once we had consistent pipelines for data curation, something that we actually could do is the analysis of this data in the continuous form to try to assess the prevalence of new variants in the society, the existence of hotspots, and this plot here on the right-hand side shows exactly that, the prevalence of the virus in different regions of London and UK in general, the effects of the vaccination, both in terms of efficacy and symptoms. But what I actually focus on, it was on the early detection of the infection and the long COVID and symptoms profiling. And the reason for that is because I was trying to monitorize and trying to describe the disease on its acute state and also then as well in its long form, basically. So at the beginning, I will start presenting a work that was published last year. It's available online, and it's basically describing the early detection of the virus. So the aim of this study was trying not only doing the diagnosis of the virus per se, but trying to also identify possible foci of infection and trying to referencing subjects, telling them you should go for a testing because there is a given probability of you being positive. So this work was developed using a Bayesian framework and the probabilistic side and the probability of being positive or negative. And basically, we used any information regarding the symptoms that the patient made available, but also the preconditions and population demographics that we had. We did this for every time point from 1 day to 3 days since the onset of the disease. So we try to do it as early as possible, again, to try to promote the referencing for testing and trying to advise for self-isolation or even just for testing. Our model was able to do the prediction independently for each one of the time points, or actually the prediction based on the 48 hours, so the first 2 days, or the 72 hours. And given this, we wanted give the outcome, but also the uncertainty that is inherent to this outcome given this probability. So the type of models that we picked was actually the Gaussian process model. And the main reasons for that is actually three. The first one is because allow us to establish the correlation between the different samples, in this case, between the different subjects and their experience with the symptoms. The second reason is because, as I mentioned, we wanted to give a Bayesian framework, a probabilistic way of measuring the possibility of being positive to COVID infection. And the third reason was actually because of the temporal component of these models. So these models are very useful to study time series. So what we tried to do, as I was saying, it was trying to measure the correlation between the subjects. And if we look at this matrix here, this is just an example. Our J here would represent the subject. So this would be the similarity measure between each one of the subjects in this big square here. And each one of the small squares would represent the correlation between each one of the time points that this subject was reporting symptoms, so DF. So when have this in mind, we have a hierarchical structure. So we want to establish correlations at different levels inside of the model. So the first one would consider, as I was saying, the patient's information. So all of the demographic information that include age, gender, and body mass index, but also the risk and exposure to the virus. And here we just used mainly the occupation of the subjects and the fact that they were healthcare workers and so they could be more exposed to the virus. So this would be our first kernel. Our second level and second kernel in this case would include all of the information related to the body mass index, preconditions that the patients would have, so comorbidities, and this includes general diseases that could emphasize the symptoms like cancer, Lyme disease, so on and so forth, but also the prevalence of the virus in a given region of UK in this case. And the last level, we wanted to model the symptoms that the patients were reporting in each one of the days across the different time points, but also how these symptoms could be impacted by the age group of these subjects. And among the symptoms, we have the list here. It's a quite extended list that goes from neurological symptoms to more gastrointestinal or even just fatigue and flu-like symptoms. So once we have that, and keep in mind what is the structure of normal Gaussian process, we just use a model with mean zero, and we encoded any type of information inside of our covariance matrix, here represented with a sigma, which is basically the hierarchical structure that I just described. So in this case, we have a covariance over the features across the time points, so between each one of the subjects, a covariance of the features across subjects, so an attempt to try to correlate the features for different subjects, and lastly, the covariance of the subjects itself. So what explains, sorry, apologies, the patient's demographics. So once we have that, and keeping in mind this is a work that was published last year, we focus our sample between the beginning of the pandemic, when we start having more data, so in April 2020 till October 2020. So we are still talking roughly about a wild type variant of the virus. And we use as training set a huge amount of data and a smaller sample for the validation, where we try to assess the performance of our models. Something that you can notice here is the fact that our data was highly imbalanced in the class. So the number of positives was much less than the number of negatives. And the reason for that is because the people in the app could test for many reasons, so contact risk, or because they were meant to, due to the job that they were doing, so on and so forth. And that's the reason of the imbalancing. The models were quite, at the beginning, hard to train due to this imbalancing, but after tuning, we reach a performance of 81 in the AUC for the 72 hours all over our population. And these are the results for the validation set. And in terms of precision and recall, we also had a good performance for the 3 days with a lower performance for the 1 day and 2 days, as you can see here. So this type of model provides not only the characterization over time, but also a prediction or a possibility to be used for patients referencing for testing. So we wanted to compare with what was available at the time in the UK. So the first thing that we call here Nhs Algorithm, it was basically the guidelines that the National Health Service in UK was giving for people to test. So if people had any of these 3 symptoms, they should be testing. And keep in mind that this was at the beginning of the pandemic. Nowadays, this list would be more extended. And we also compared with another model, a logistic regression model that was used using the peak of the symptoms at the time, and basically was using the same data that we had. So it was the first attempt of characterizing the disease with our sample. So I will try to go through this table. I realize that it's quite massive. So what we can see here is for the 3 days, our model is consistently better in terms of ROC AUC when compared with the previous model, the logistic regression that has been developed by many et al., but also better in terms of performance and accuracy than the Nhs algorithm. When we look at specificity, we can see that actually our model performs in a worse way and captures more false positives. But this was not a problem, or it was not a concern for us, because actually, since we were not using it as a substitute to PCR, but rather just an indication that maybe the subjects should do isolation or testing, we were more concerned about the sensitivity. So we wanted to capture as much as possible the true negatives, even with the issue then including some false positives. And this is what we can see here, that in terms of sensitivity, our model outperforms the other 3 algorithms that were at the time being used. So another thing that we wanted to explore was the use of this type of Bayesian models to characterize subpopulations or strata of the population. So is the model in the same way efficient in different population strata, or are the features that are relevant to the model the same across different populations? So here in this plot, what you can see is our analysis in terms of age groups. So the black gray that you see in all of the plots is basically the features relevance across when trained the model with all of the population, so across the full population. And then we have in which are not of these plots for subgroups of the population. And something that we found quite interesting is that the symptoms that are more relevant to early detect or to detect the early signs of the disease across younger people, so 16 to 39, is actually the loss of smell. They are mainly neurological, with some incidence of chest pain, for instance. But these symptoms are quite different when we look at older patients or older subjects, where doesn't mean that they wouldn't have loss of smell or any other respiratory symptoms. It just means that the early detection was mainly based on gastrointestinal symptoms, for instance, and other types of symptoms like flu-like, and this is quite interesting to see, at least in our population in UK at the time. So we did the same kind of analysis between genders, and here we have women and men, and we see that we have a wider range of symptoms reported by men that are relevant to the detection. But I would like to focus on the occupation, so the risk and exposure to the virus. And something that we see is that the fact that for the normal population, which was the one influencing the model the most in the end of the day, because it's the bigger sample that we had, the symptoms are exactly the same as we expected. But then for the healthcare workers, so this plot here, we see that they show as well a wider range of symptoms. So our belief is that since the healthcare workers were contacting more closely to the virus, they were more aware of other symptoms that were not as common or as mentioned in the media at the time. And they were reporting those as well, which was influencing the early detection of their disease. So as I mentioned at the beginning, we also wanted to look at the uncertainty of the predictions. Okay, the model might be right, but how certain is it about the prediction that it's doing? So what we can see here is basically the likelihood of the predictions that we have. So basically, the wider the distribution, more uncertain is the model. And something that we can see, interestingly, is that the differences between 2 days and 3 days of information in terms of certainty of predictions are not that big across the group, across the different group age. But when we look at the older patients here, we can see that actually having 3 days of information actually improves much more the performance of the model rather than just having 2 or 1 even. Which indicates, for instance, then maybe their symptoms are more uncertain, or maybe the model needs more data for this subset of the population to be as accurate as for the rest. So we did the same kind of analysis again for the same substrate of the population, so female, male, and also healthcare workers, non-healthcare workers. And if we see here, the most uncertain one is actually the healthcare workers. And we believe that the reason for that is exactly the fact that, first of all, there were a small sample of our overall population, so the model had less data to be accurate on those. And on top of that, they had a different subset of symptoms. So they were reporting differently from the overall population, hence the fact that the model was not as accurate as it should for these patients. So in terms of take-home message from this study, something that we were quite happy about it was the fact that we had the opportunity and we had the chance to show that these population-based studies with these large data sets actually are very informative, and making use of the general population and not just a research trial that has been designed specifically for a disease can still inform and give some insights over the disease itself. Another thing, it was the fact that these kind of models in specific can be highly useful, not only in this pandemic, but even in other diseases or other pandemics, to detect the early signs of the disease and actually to provide some referencing for testing and some support in terms of guidelines and management. In terms of limitations, as you can guess, the main limitation is the fact that these are trainable models to a specific population, which means that, and in specific, they are data-driven for these symptoms, which means that if we had the phenotyping changing a lot due to new variants or vaccination campaigns that might change the symptoms reporting, the model would need to be trained again. And this one might be less accurate than is currently for the specific population that is being tuned. And this brings us to our last limitation of this model, which is for the ones that already have worked with Gaussian process. They are nice models, but they are quite hard to optimize for big data sets due to the optimization that is in the back end of these models mathematically. So they are very computationally expensive. And as a consequence, it might take a while and days, or it might require a huge memory in terms of GPUs and computers to actually do the optimization. So it's something that cannot be continuously done or online or not at the stage that has been developed at this point. Nevertheless, we still believe that it was quite useful to capture the temporal pattern of the symptoms, and it was very successful in search early stage of the disease. So for the second part of the talk, I will be talking about the profiling of the post-COVID syndrome. So this work is also available online in medRxiv, and basically just tries to explain if there are different profiles for the symptoms of post-COVID syndrome, and if they exist, if they are different or not across variants of the virus. So when a person is infected by different variants. So we define here post-COVID syndrome as persistent symptoms for longer than 84 days. And we use, again, the information reported in the app, both the systemic symptoms that the patients were reporting, but also the demographic information. So our pipeline in this case, it was acquiring all of the available information that we had for patients that have data for more than 28 days, and then analyzing for the ones that had more than 84. We avoided any missing data for more than 7 days because we wanted to be sure that it was still the same disease going on and not just the symptoms stopped and the patient is feeling something different, and it wouldn't be post-COVID syndrome in that scenario. Once we have that, we did an aggregation in terms of weeks just to reduce the amount of data because it would be a very long time series analysis if we keep the records for each single day. So we did the cumulative sum of the symptoms per week. And then we did the clustering optimization over the time series that we had. And once we obtained the clusters, we tried to do the characterization of them in terms of profiles of symptoms. So the model that we work here, in this case, it's not original as in the early symptoms detection. So this model is actually an adaptation from a previous work from Liane et al., also available online. And what they did was actually trying to characterize each one of the patients throughout a covariance matrix. So again, establishing the correlation between the symptoms over the time points. And this brings us to a covariance matrix that it's basically just if F, it's the symptoms that we are considering, all of the symptoms by all of the symptoms across the time points. The very useful thing about this model is the fact that we can have, and here we have the example of 2 subjects. We have actually subjects with different lengths for the time series reporting. So a subject might have long COVID for 12 weeks, another one might have for 15 weeks, and the model can still cope with that. So once we have that, if we imagine that we have groups of subjects, we can average their covariance matrices describing the features over time, so the symptoms over time, and create templates. So using single value decomposition, which is like a PCA, we can basically characterize their evolution of symptoms over time as a template for that specific group of symptoms. So if we define a given number of these template matrices, like K, being K the number of clusters that we have available, and then we perform K-means, we can actually do the clustering of the patients giving this time series that we have. So then the question is, how can we decide the number of clusters? And in this specific case, we wanted to be sure that we actually had the correct number of clusters to characterize the disease. So the way that we did it was through a Bayesian information criterion approach, where we just pick the minimum number of clusters that achieve the lowest error. So this is the plots that you can see here. And we run this analysis for 2 to 15 clusters. So basically, we just optimize the model considering that we would have 2 clusters, 3 clusters, so on and so forth, to 15. We also did a bootstrap analysis to just be sure that our model, or at least the number of clusters, would be robust to different populations. So in this case, as I was saying, we used the CSS, so the app data, but we also used this sample here called CSSB, which was basically a subsample of the original population that also did a questionnaire, an online questionnaire, describing their life experience with the virus in the long COVID form. So basically, they just reported how did they feel in terms of the impact of long COVID in their lives, if they have been hospitalized, so on and so forth. So we did the analysis in a split manner. So we tried to analyze the unvaccinated versus vaccinated because we assumed that the profiling of the symptoms would be different, not only for the variants, but also if the patient had the infection before or after vaccination. We know that the ratio of infected people for vaccinated, the ratio of long COVID among vaccinated people is actually lower. So we wanted to actually disentangle that. And we split it in this manner. And we did it for the main variants in the UK. So wild, alpha, and delta types of the variants. Unfortunately, we didn't have enough data to do the unvaccinated in Delta, so 132 to the clustering analysis, given the number of symptoms that we had was insufficient. In the same manner, we couldn't perform as well for the vaccinated in wild and alpha, because again, it was quite reduced. So we ended up doing the characterization of wild variants, of variants for unvaccinated, and Delta variants for vaccinated people. So the way that we picked which variant had contaminated, since we didn't have genetic information, but just the self-reported tests, was based on the prevalence of each one of the variants in the population in the UK. And we just selected a threshold of 80% among the population to then assign the variants to each one of the subjects. Once we have done that, we optimize the clusters. And for instance, here we can see the obtained profile for the wild type variants. And something that we can see is the symptoms. So here we have the week of the disease, and this color map highlights the ratio of subjects reporting each one of the symptoms in which one of the weeks, the ones that were contaminated with the wild type variants. So the darker, the more patients were reporting this symptom in this given week. And we can see that the symptoms were quite different across the profiles, but we wanted to quantize this difference and actually have a better profiling in terms of the symptoms, duration and prevalence. So what we did was a Z-score quantification comparing each one of the patients inside of a cluster to the general population infected by these variants. And this gives these matrices that we have here, where basically the red encodes the most relevant symptom that differentiates this cluster from the remaining four. So it doesn't mean that is only the most relevant for these patients. It means that is the one that allows us to differentiate this cluster from the other ones. And once we have done that, we could build these plots that highlight for the third quartile of the highest Z-score, so the really ones that are the most relevant across the time series, which ones characterize each one of the clusters. And we can see that in terms of profile, so here, the width represents the symptoms prevalence reported by the subject. So how many subjects, the median of subjects that reported the symptom across the full time series, whereas here we have the Z-score value. So we can see that the different clusters actually have different influence of the group of symptoms that we can see. With the cluster A, for instance, have mainly cardiorespiratory. This is also the biggest cluster that we had. And then we have the smaller cluster with being affected with the majority in terms of central neurological, abdominal, and also upper respiratory, but not the cardiorespiratory. So the main conclusion from here is the fact that, yeah, it exists different profiles of the disease among the wild variants. Can we replicate this for the other variants? So we did the same kind of analysis. In this case, for the alpha, we had 7 clusters. So the main reason for that is because sample was slightly smaller than the rest of the variants that we had, so the model struggled a bit more to optimize. Nevertheless, we replicated exactly the same results. And we had the same kind of scenario. So for instance, for the biggest cluster, we had a predominance of central neurological symptoms without much influence of the others. Whereas for the smaller cluster, which we characterize as the most heterogeneous one, we don't even have this group of symptoms anymore. And once again, this showed that even though these clusters were a bit more fuzzy and not as well defined as for the wild, we still see different patterns of symptoms across them. And lastly, for the Delta variant, we did the same analysis. In this case, we find 5 symptom clusters. And once again, we see the same kind of behavior. So we see the biggest cluster with the majority of central neurological symptoms. And for the rest of the clusters, we can see that sometimes we don't even have the influence of this group of symptoms. And the smallest one is once again the most heterogeneous one. So once we have done that, and keeping in mind that we had this subpopulation that answered a questionnaire about their life experience, we wanted not only do the profiling of the symptoms, but see if these clusters in specific with these given symptoms would have a specific impact on the people's life, basically. So in this case, we only did this for the wild type variants, and the reason for that is because it was the only subsample that actually answered this questionnaire. It was released during this time. And we focus on specific questions that translated the impact. So people that look for medical help at the acute stage, people that look at medical help during the prolonged disease, hospitalization, reinfection, and if they had severe impact on their daily activities or just prolonged impact, so on and so forth. So something that we could see here is the fact that the one that caused more search for medical help during the course of the symptoms was actually cluster B. And if we look at the cluster B again, we can see that it's the one that it's linked to systemic inflammatory and immune-related symptoms. So this is quite interesting because it means that we can not only do the profiling of long COVID, identify different patterns of symptoms for long COVID, but actually see the meaning of this in the daily life of these patients, of these subjects. Similarly, we also had some impact of cluster C and D, for instance, on the severe impact on their daily lives. And once again, it's linked to upper respiratory and system inflammatory thing. And yeah, this can be used in an efficient way. So this is basically just a logistic regression analysis with odds ratio where red actually encodes significantly differences that we can find here. So the main contribution of this work in our perspective is the fact that, yes, we can tell to the clinical teams and the clinical context that exist different profiles of symptoms for post-COVID syndrome. So it's not a homogeneous disease. It doesn't mean that the patient has long COVID, it will have the same symptoms than other patient, or they will have the same duration. We can actually see changes in terms of that. But more interesting than that is the fact that we have 3 major clusters that are replicated across the 3 main variants. This means that in each one of the variants, we will have a group of patients that show mainly central neurological, and interestingly, across patients with alpha and delta, this is always the biggest cluster. But then we also have a cardiorespiratory cluster and a systemic and inflammatory that actually usually it's the smallest one across the 3 types of the virus. Another thing that we can conclude from here is the fact that maybe, as we did for the early diagnosis analysis, is the fact that having a subdivision and sensitivity analysis on the population strata in terms of demographics could be interesting to see the impact of these clusters on that. We have in our study online available some analysis in terms of demographic profile, age, gender, so on and so forth, but maybe an optimization of the clusters having these covariates as symptoms as well would be interesting. In terms of limitations, is the fact that we don't have genetic information about these patients. So we are talking about the phenotype, but we never characterize the genotype of these patients in terms of the long COVID that they show in terms of symptoms. And we also didn't have a chance to do any analysis regarding the vaccination in terms of timing, doses, vaccine type. So we tried to separate the groups in terms of unvaccinated or vaccinated before the infection, but still a more sensitivity analysis should be conducted on this regard. And lastly, we couldn't also do it this for Omicron because we didn't have the data to support the analysis at the time that it was conducted. But as a take-home message, we believe that this is really relevant because once more, and similarly to what happened to the early diagnosis part, we can use data-driven approach and population-based studies to actually give some insights about the disease that is just new and it's just we don't know much and we actually want to learn about it. So this kind of models has shown to be robust and very interesting to provide as much information as we can get at this stage. So as you can guess, this work I'm one person. I'm just the face of all of this work. This couldn't be done without all of the collaborators. Some of them are here listed, but there are many others that have helped throughout these 2 years to conduct studies. And of course, I would like to thank as well to the study participants, because without them, we wouldn't be able to do this analysis for sure. So let me know if you have any questions, and thank you again for this opportunity.
Thank you so much for the great presentation. Do we have questions?
Sorry, I have one.
Yeah.
Yeah. Well, first of all, great talk, and thank you very much. It's very interesting. So mine is very much at the beginning, and I might have missed that. Is you essentially use a continuous model for both your stories, and you use self-reported symptoms. So some of those, for example, in this graph where you show the diagrams, and I wonder, there are two things. One is there are some that are like a little bit binary, like I have CAF or I have no CAF, or they might be translated into I have a CAF 1 or I have a CAF 5. So one question is how do you cope with categorical variables, if you did include them or if you just made them continuous? And the second one will be how, if there is some part, some pre-screening of the data or some pre-processing of the data regarding the intrinsic uncertainties of self-reported symptom. Let's say, well, fever is very clear, but CAF, again, if I quantify that, I might think that I have a CAF 3, and someone else might say, no, it's a CAF 2 or it's 4, etc.
No, definitely. I will start with the last part. So in terms of data curation, we tried as much as possible to remove any bias or any source of error inside of our data. So we started removing any kind of reporting that was not consistent over time. So people that report wrong BMI or wrong age, or even that the report was not consistent, we removed those to try to reduce the amount of noise that we could have. Then there is a source of bias that we cannot remove, which is the comparison between the symptoms reported across people. We cannot be sure that the 3 of cough is actually a 3 and not a 2, and so on and so forth. And it's one of the limitations of our studies. The advantage of the fact of being a big study is the fact that we try to homogenize the impact of such differences with the high numbers that we have. So it's normal that the perception of the symptoms are different, but with such big numbers, this should be homogenized itself with the data that we have. And if you think about it, it's the same thing when a clinician takes a patient and asks for, how do you feel? How is your pain? It's a matter of perception. So since we are trying to tackle what is the perception of your disease, it's a bias that we acknowledge, but it's not as severe as it could be. Now, going back to your first question, that's a very good point. So for the second model, actually, we binarize all of the symptoms to yes and no. So when we had a scale, we had severe fatigue, mild fatigue, and medium fatigue for the scale that we had, but as a binary. And for all of the others, we just had yes or no. Fever, yes or no, given a threshold, so on and so forth. For the first model, though, which is the one that I have the slide here, we actually consider the scale. And the reason for that is because Gaussian process has something called RRD, which basically is automatic relevance determination, which allows us to optimize this L here, which is a length scale for each one of the features, which means that this has been optimized for each symptom individually. So when we concatenate the matrix, this is already been considered by this length scale. And doesn't matter if the scale for one is between 0 or 3 or it's between 0 and 1, because when they are concatenated, this is already encoded by the kernel matrix that was specifically for that symptom. So that's another advantage technically wise of Gaussian process. It's the fact that we can do just then the multiplication of these kernels and put them together, but we can optimize this cell separately for each one of the features. It's because that we can optimize, and I'm sorry, maybe I went too fast on this and I didn't go into the detail that I would like, but this relevance that we can see here, it's a relevance that it's obtained based on this length scale that we extracted from the Gaussian process per symptom. So the length scale is basically a measure of how wide is the window that I'm looking at when I'm seeing the symptoms. So how granular is the information that I'm retrieving from them? And based on that, we can use it as a surrogate measure of the relevance. Because if this length scale is too big, it means that all of the information is relevant. So there is no peak, no relevant part that actually should be retrieved. If it's small, it means that there is lots of granularity that's actually relevant, which means that that symptom is specific. It's providing lots of information. We try to describe that in the paper, but yeah, it's like that. How do we encode different scales of the symptom in this specific study? In the second is as simple as binarizing everything, because we just computed the covariance matrix without the optimization of these parameters, basically.
Thank you. It's amazing.
Other questions? So I'm sorry if I missed that, but the data, are the data publicly available to the community yet, or?
Yeah, so at the beginning, we made the effort to make the data available. I'm not the one coordinating the acquisition of the data. That's on the hands of SOE Health. And to be honest, at this point, I'm not sure how available it is publicly. We have in our papers always the directory, the online folder where you can access the data, but I would need to double check if it's still online and available, because yeah, as I was saying, this was a partnership between King's College and other universities, actually Harvard and MIT as well, but the data was mainly managed by Zoe Health, so okay. I would need to double check that.
Okay, thank you. And related to this, and maybe do you know if the data are available, like when they will be available, but do you have them in a sort of standardized format that once they are available, they would be easily processed and further, analyzed by others?
Yeah, definitely. So the data, it's recorded as a CSV file. It's a massive CSV, so we convert it into other forms. This etc. Software that I just mentioned, if you go to the GitHub page, and I can go back and just show the GitHub page again if anyone is interested to double check it. It's here. You can download the schema that explains you how the data is organized. You actually have all of the keys of the data, so on and so forth. The format, it's called HDF5 file, which is a different format to just save some memory. But by downloading the software and using that structure, you can easily read the data. In terms of using the data, I'm pretty sure that if you contact the PIs of the project, and we are talking about Sebastian Arsalan or Claire Steves, I'm sure that they would appreciate people interested on these results and would be happy to work with people. So if anyone is interested, just let me know or contact them directly, and I'm sure that it would be easy.
Okay, thank you so much.
Right.
Okay, we have another question there.
Yes, thank you for the interesting talk. So this was about what you presented was about the early variants, the wild type, the alpha, and the delta variants. And as we know, since the advent of Omicron is not a single thing. There's a whole like phylogenetic tree of many variants. So is this ongoing, and is it tracking like all the multi-numeric classifications of different variants that are existing now?
So the study is still ongoing. So if people want to participate, they can still log their data in the app, and the data then arrives to us and we can process it. About the subvariants, something that we need to keep in mind is the fact that we don't have the information about the subvariants per se per person. So we have the subvariants in terms of prevalence in the region, in terms of the society, but we don't have the PCR outcome. So we are never sure which variant each person has, just to keep that in mind. Now, answering to your question about could we do this new analysis in these subvariants and try to see how different they are, we could. The major problem of that is the fact then with the less restrictions and with the time, people were less prone to actually report their symptoms and be concerned about COVID. So more and more, we have less data and as a consequence, less reliable data in that sense, because people are also less concerned about COVID. And this is a consequence of the fact that it's a study population, and basically we cannot control how many contributors we have. But yes, even with our numbers, we could still replicate all of this analysis and see how does it behave for Omicron. We haven't done it. We have just tried to make available these papers, but it's something that we can conduct. And if people keep reporting, which we hope that they do, we can even evaluate further along the impact of the pandemic, even years to come, which would be really interesting in terms of sequelae and post-COVID syndrome for sure.
I wonder if there could be a dynamics of the sampling bias in that people who have symptoms are more likely to report, like people who have serious symptoms are more likely to continue to be motivated rather than people who have mild symptoms that they are, they want to forget about COVID.
Yeah, you are 100% right. And we have other studies that I didn't mention here because I was not the leader in that studies that assess exactly what you were saying. So we had a study that assess how prone a person is to take a test, given their demographics, their background, the type of symptoms that they had, if they had a previous infection or not. We also had another study quite interesting about the impact of the pandemic in the mental health and how that does impact as well the reporting of the symptoms per se. And we are about to release as well another study regarding the symptoms reporting and how different they are before and after long COVID syndrome. So this is ongoing work that it's still being conducted, although it's already published. So if you have the chance to look at the data and online in Scholar, anything that is reporting in Scholar and Zoe addresses some of the topics that you just mentioned. And we have seen it, of course. People that experience more severe symptoms are more prone as well to keep logging them and keep the track of such symptoms over time. And that's actually the reason why we have such a good sample for long COVID as well, because people keep reporting because they still feel sick. Yeah.
Right, but then it becomes difficult to have like matched controls. But anyway, yes, thank you.
Yes.
Any other questions?
Well, we'll have a last one if nobody else. Otherwise, I will give road.
Go for it.
I go. So, well, mine is, let's say, probably we go to at the end. Have you found or have you built purposely, perhaps I missed that, in your second study, I think, some statistical model that gives some suggestion according to a certain, I don't know, you find that the patient pertains to a certain cluster and we should care a little bit more, or even if with mild symptom, we should check it more than some other patient, also including all the other comorbidities, other stuff, cancer maybe, and stuff like that. Is there any prognostic value that you can add to the model?
So no, the short answer is it's not right now. So I'm trying to move. Yeah, we did some sorry, apologies for that. So I'll just come back and present again. So maybe I can show the screen. Can you see the screen? Okay, so we had some individual analysis of the clusters. So we try to project what would be the symptoms of each one of the clusters individually. Here, I'm just taking the example of alpha. Again, this is in the paper, I just didn't present it here. And we try to see if these symptoms replicate individually and what is the impact in their lives. So that part we did. In terms of preconditions, we also try to see the incidence of each one of the preconditions, also in the paper, for each one of the clusters. But we didn't include that in the analysis. That's something that we are looking for to do as a future work, to try not only model the symptoms per se, but what is the impact of each one of comorbidities in terms of the profiling, but not at the moment. Not for sure. And the main reason for that is because when we were building the covariance matrices, we wanted to focus only on the symptoms, and we didn't want to put any confounding effects on the preconditions on top of the symptoms. So we wanted to just see, are the symptoms sufficient to identify profiles, rather than, okay, let's dilute a bit the impact of the symptoms given some preconditions on top of that. That's the main reason why we went for the simple approach. But at least some measurements in terms of statistical analysis are in the paper, just reporting what is the distribution of preconditions per cluster across the variants. But I agree with you. That's a very exciting new work, which is modeling the profiling given symptoms, but also giving all of the demographic information that we have about the patients. Regarding the genetic part, we don't have that information, unfortunately. Regarding the hospitalization, we only have that information if the patient reports. And we are quite cautious about the hospitalization per se as well, because it's a biased information. As the previous person was saying, our data is biased, and if a person feels more symptoms, might report more, but if a person goes to the hospital, might not report that because it's hospitalized. So we are quite cautious in the use of the hospitalization information because of that. And since we cannot link the data from the app to the health records from the hospitals because of anonymization issues, we cannot accurately perform that analysis as well. If that makes sense. I hope that I explained well.
Yeah. Thank you.
Yeah, no worries.